Hello, greetings, and good day. So for this last installment of our online lecture on the circulatory system, we will be zooming in on the fluid medium, which is the blood. Now, what is in blood anyway? Now, our blood is a homogeneous mixture of the following components such as red blood cells, then we have platelets, also we have white blood cells, and we have carbon dioxide, digested food, then we have plasma, which is the liquid component of the blood, dissolved hormones in the blood plasma, oxygen, and waste products like urea. So our blood is a homogeneous mixture of those components. So we have the red blood cells, white blood cells, and platelets. Those are the solid components of the blood. And we have the liquid component of the blood, which is plasma. Now, if you will place blood in a test tube and run it through a centrifuge machine, you should be able to separate the components of the blood. So we have here the yellowish straw-colored liquid, that is the blood plasma that carries the blood cells and platelets. Then we have beneath the plasma is a buffy coat that contains white blood cells and platelets, and below the buffy coat is the solid component of the blood, which are the red blood cells. Now, plasma contains the following. We have dissolved gases like carbon dioxide and oxygen. We have also glucose, amino acids, proteins, minerals, vitamins, hormones, and waste products like urea. So that's the liquid component of the blood or plasma. Now, suspended in plasma are the solid components of our blood. So we have the red blood cells, white blood cells, and platelets. So let's start now with the red blood cells, also known as the erythrocytes. Now, under the microscope, red blood cells or erythrocytes, they are biconcave disc, more or less like a donut. And these red blood cells or erythrocytes, they are produced in the red bone marrow. And they are named red blood cells because of its reddish pigment or hemoglobin. Red blood cells contain hemoglobin, which is a molecule specially designed to hold oxygen and carry it to the cells that need it. And also red blood cells, they are capable of changing shape to an amazing extent without breaking, as it squeezes single file through the capillaries. So those are the red blood cells. And the fact that red blood cells are denucleated or they don't have any nucleus, they have a limited lifespan of only 120 days. That is the reason why the red bone marrow must produce millions of red blood cells each second to replace those red blood cells that have died out. So isn't that fascinating indeed? Now let's proceed to the next solid component of the blood, which are the white blood cells or leukocytes. So these leukocytes, they are fewer in number, they are bigger than the red blood cells, and they contain a big or large nucleus. And there are several types of leukocytes or white blood cells. So these are the lymphocytes and the macrophages. Both lymphocytes and macrophages, they play an important role in our immunity. The macrophages, basically they eat and digest any microorganisms that might have invaded the blood. And lymphocytes, on the other hand, they fight disease by making antibodies to destroy invaders by dissolving them. Other lymphocytes, they make antitoxins to break down poisons. Now, aside from classifying white blood cells based on their role that they play in the immune system, we can also classify white blood cells based on their appearance under the microscope. So we have what we call granulocytes and agranulocytes. Now, granulocytes, these are white blood cells that contain granular cytoplasm. And agranulocytes, these are white blood cells that doesn't contain granules in their cytoplasm. Now, under granulocytes, we have the following types of white blood cells. We have neutrophils that helps in phagocytosis or cellular eating. Then we have eosinophils that fight against parasitic microorganisms. And we have basophils that play an important role in allergic reactions. Then we have the agranulocytes. Under agranulocytes, we have lymphocytes that produces specific antibodies. And we have monocytes that fight out bacteria, viruses, and fungi. So those are the types of white blood cells. Now, at this point, let us now differentiate between neutrophils, eosinophils, and basophils. Now, if we will look closely at our illustration, neutrophils under the microscope, they have a multi-lobed nucleus. And they engulf bacteria found in the extracellular matrix through phagocytosis or what we call cellular eating. And they account for 40 to 75% of the total white blood cells. Then we have the eosinophils. Eosinophils, they are the second type of granulocytes found in our blood, and they are involved in triggering inflammatory responses during allergic disorders. And eosinophils, they also combat multicellular parasites like helminths, responding to chemokine and cytokine signals. Eosinophils migrate into inflammatory tissues. And lastly, we have the basophils. Basophils under the microscope, they have a bean-shaped nucleus. And they contain heparin, which prevent quick blood clotting. And they account for about 0.5 to 1% of the total white blood cells in the body. And lastly, we have the so-called platelets or thrombocytes. Now, actually, platelets are not cells in the sense, but these are cell fragments. And platelets, they produce tiny fibrinogen fibers that forms a net. This net helps trap other blood cells to form a blood clot. So these thrombocytes or platelets, they play an important role in blood clotting. So before we proceed to the cardiac cycle, let's watch this fascinating video about the blood. Alright, let's watch this.
Of all the substances within the human body, blood is one of the more familiar ones, as we've all had an injury that involves bleeding. For a long time, it was not well understood exactly what blood is, or what it does. We just knew that if you lose enough of it, you die. But we now have an intimate understanding of this fluid, as well as the circulatory system, whose function it is to continuously pump blood around the body. We will get to this system in a moment. First, let's examine blood itself, what it's made of, and why it is so critical for human life. First, let's mention that blood is technically considered a connective tissue, and as such, it is the only fluid tissue in the body, full of fibrous proteins. It is comprised of formed elements, which are blood cells, suspended in a fluid called plasma. If we place blood in a centrifuge, it will separate into its components. The densest section is comprised of erythrocytes, or red blood cells, and the least dense section will be the yellowish plasma. They are separated by the buffy coat. a white layer containing platelets, as well as leukocytes, otherwise known as white blood cells. As a whole, blood is responsible for distributing various substances around the body, most notably oxygen, which we can't survive very long without. But it also carries nutrients absorbed from the digestive tract and hormones secreted by endocrine organs. Blood also delivers waste products to the organs that will dispose of them, like the carbon dioxide that we exhale. Beyond this, blood serves to regulate pH in various tissues, maintain body temperature, and prevent infection. Let's discuss each component of blood now, beginning with plasma. This is a sticky fluid made mostly of water, but also containing a variety of proteins, nutrients, ions, gases, and hormones. The most abundant plasma protein is called albumin, which contributes significantly to plasma's osmotic pressure, and this is followed by a variety of globulins which bind to certain molecules for transport. Moving to the formed elements, these are erythrocytes, or red blood cells, leukocytes, or white blood cells, and platelets. Red blood cells and platelets are interesting in that they don't possess all the typical organelles and they don't divide. They are replaced by stem cells in the bone marrow. Red blood cells are very numerous in the bloodstream, and they are shaped like flattened discs with depressed centers. There is no nucleus, not much of anything inside other than lots of hemoglobin. This is the protein that allows for the transport of oxygen throughout the bloodstream, which is picked up in the lungs and then released for tissue cells throughout the body. There are other proteins as well that have structural or protective functions, but hemoglobin will be the focus here. This is the structure of hemoglobin. It is made of a protein called globin, consisting of 4 polypeptides, 2 identical alpha chains, and 2 identical beta chains, each of which is bound to a heme group with iron at the center. The iron in this heme is able to bind to an oxygen molecule in reversible fashion so that it can bind and then release when necessary. So each hemoglobin can bind 4 oxygen molecules. And there are around 250 million hemoglobins per red blood cell, so one red blood cell can transport 1 billion oxygen molecules. Blood cells are produced through a process called hematopoiesis, and this occurs in the red bone marrow, which is a soft network of connective tissue found on certain blood capillaries, and which contains hematopoietic stem cells. For erythrocytes, this is more specifically called erythropoiesis, and billions of new red blood cells are made every day to maintain a nearly constant number, given that red blood cells function properly for only about 3 months, only to be destroyed by macrophages, which are a phagocytic type of white blood cell. Next, let's look at leukocytes, or white blood cells, which, unlike the far more abundant red blood cells, are complete cells with nuclei and organelles. These are part of the immune system, which we will discuss in greater detail later, but for now, we will just understand that these help us defend against pathogens and other harmful things. These use the circulatory system to get around the body, but they can also slip out into other connective tissues to do their work. There are 2 types of white blood cells, granulocytes and agranulocytes, which differ in the presence or absence of granules. The 3 types of granulocytes are neutrophils, which kill bacteria, eosinophils, which kill parasitic worms, and basophils, which contain histamine that attracts other white blood cells to a site of inflammation. Agranulocytes, on the other hand, can be lymphocytes, which fight viruses and tumors, and also give rise to plasma cells, which produce antibodies, which we will discuss later. or they can be monocytes, which become macrophages that can eat up intruders. Leukocytes are produced by leukopoiesis, which is stimulated by certain chemical messengers. Lastly, we get to the platelets. These are fragments of large cells called megakaryocytes. These fragments are essential during blood clotting, which happens when blood vessels are damaged, as platelets can plug up any holes or tears to seal things off. They flow through the bloodstream in an inactive state unless needed, dying every 10 days or so and constantly regenerated. Megakaryocytes form due to repeated mitotic cycles that do not perform cytokinesis, so the result is one huge cell with a multi-lobed nucleus. This then presses against a sinusoid and its extensions burst to release the platelets. These platelets are important during hemostasis, which is the process by which the body will stop bleeding through vascular spasm, platelet plug formation, and then coagulation, or blood clotting. This essentially means that where there is damage to a vessel, smooth muscle will contract, platelets will plug the tear, and a protein called fibrin will form a mesh to patch everything up. Once the vessel has healed, the clot is removed through a process called fibrinolysis, so that there is no blockage in the vessel. So those are the components of blood.
Alright, so that was a very interesting video about the components of our blood. So I hope you have learned a lot through that video. Now at this point, let us now study the so-called cardiac cycle. Now the cardiac cycle is composed of the so-called systole or systole and diastole or diastole. Now, systole or systole in the cardiac cycle refers to the contraction of the ventricles. And the contraction of the ventricles will lead to the opening of the semilunar valves that forces the blood into the arteries, either the pulmonary artery leading to the lungs and the aorta. While, on the other hand, when we speak of diastole, this refers to the filling of the ventricles with blood. Diastole in the cardiac cycle is the period of relaxation of the myocardium or the heart muscle. And this is accompanied by the filling of the chambers with blood. Now, to help you visualize the cardiac cycle, let's take a look at the following diagrams. So in systole, the ventricles contracts and this will open the semilunar valves and the blood is forced either to the pulmonary arteries leading to the lungs and the aorta. Whereas in diastole, there is the relaxation of the myocardium and there is filling of the chambers of the heart. So the ventricles are filled with blood and the atrioventricular valves are open. So again, when we speak of the atrioventricular valves, these are the tricuspid and the bicuspid or the mitral valve. So that is the cardiac cycle, systole and diastole. And here is an animated illustration of the cardiac cycle. So in systole, you have the contraction of the ventricles and this will open the semilunar valves, forcing the blood either to the pulmonary arteries leading to the lungs and the aorta. While in diastole, there is the relaxation of the myocardium and the ventricles are filled with blood. So that is the cardiac cycle. Now, we should also bear in mind that associated with the cardiac cycle is that how our heart sounds. Now, our heart sounds something like this. Alright, did you hear that? So our heart sounds loved up are associated with the closing of the heart valves. Now the first sound or the lub sound occurs as the atrioventricular valves closes, and this signifies the beginning of systole or the contraction of the ventricles. While the second sound or the dup sound occurs when the semilunar valves closes at the beginning of ventricular diastole or the relaxation of the myocardium. Alright, so at this point, let us now watch this video about the cardiac cycle. Alright, let's watch this.
Ventricular contraction causes the atrioventricular valves to close, which signals the beginning of ventricular systole. The semilunar valves were closed during the previous diastole and remain closed during this period. Continued ventricular contraction increases pressure in the ventricles above the pressure in the aorta and pulmonary trunk, causing the semilunar valves to open. When the ventricles relax and their pressures drop, blood flowing back toward the relaxed ventricles causes the semilunar valves to close, which is the beginning of ventricular diastole. Note that the atrioventricular valves remain closed. When the pressure in the ventricles becomes lower than the pressure in the atrioventricular valves open and blood flows into the relaxed ventricles. This accounts for most of the ventricular filling. The atria then contract and complete the ventricular filling.
Alright, so I hope you have learned a lot about the cardiac cycle through that video. Now, before we end with this online lecture on the cardiovascular system or the circulatory system, I just would like to point out one major concern related to the cardiovascular system, and that is having high blood pressure. Now, what is having high blood pressure anyway? Now, actually, high blood pressure is a sign that the heart and blood vessels are being overworked. So take note of that. And if left untreated, the disease can lead to arteriosclerosis or the hardening of the arteries and congestive heart failure. Now, heart disease contributes to 75% of all heart attacks and stroke. And it is very important to monitor our blood pressure. So let's watch this video about manually monitoring blood pressure using the sphygmomanometer. Alright, so please listen to this and this might save your life. Alright, let's watch this.
First, I clean my hands. I align the artery index on the cuff with the brachial artery and then wrap the remainder of the cuff around the patient's arm and secure the Velcro. I initially measure systolic blood pressure by palpation. I close the valve by turning clockwise and then quickly inflate the cuff until the brachial pulse disappears. I slowly deflate the cuff at approximately 3 to 4 millimeters of mercury per second until the pulse returns. This is the systolic blood pressure. I then fully deflate the cuff. I then perform a more accurate estimation of blood pressure by auscultation. I place the stethoscope over the brachial artery and hold it in position using 2 fingers. I now inflate the cuff until the pressure is approximately 30 millimeters higher than the initial systolic reading. I slowly release the pressure in the cuff while noting the point at which I hear the first pulse sound. I continue to deflate the cuff and note the point where the pulse sound ceases. I tell the patient the result, remove the cuff, and clean my hands.
Alright, so I hope you have learned a lot about manually monitoring or measuring blood pressure using the sphygmomanometer. Well, I guess that's about it for our online lecture on the cardiovascular system or the circulatory system. Please like, share this video so that we could reach more students, especially the life science students out there. And please subscribe to my YouTube channel, The Science Guy, Professor June Cajigal. And please like my Facebook page, The Science Guy TV Kaspacer channel. So this is Professor June Cajigal saying, live long and prosper. Always stay safe and so long for now.